So you have done your RNA sequencing, performed your RNA sequencing data analysis, and have identified genes that are differentially expressed. And in your analysis, you have realized that there are genes that are upregulated and there are genes that are also downregulated. What next after this? What do you do with your RNA sequencing data? Now, this is where downstream data analysis or processing comes in. And in some cases, you will have to go deeper into wet lab and look into what really happens to these genes and whether their reversal can have anything to do with the processes within your cells. I mean, this is the entire beauty of combining both the wet lab and the dry lab aspects of biology and science. So in this video, I will show you how to go deeper into your data and make deeper insights and even meaningful hypotheses and suggestions. So one of the things you can do is to see whether in the literature there are any drugs that have been published to target either your upregulated genes or some of your downregulated genes. By this, I mean finding drugs that have already been tested in other cell lines or even in your own cell lines that you can use to inhibit the proteins expressed by those genes. You know that by inhibiting the protein, you can sometimes reverse the cellular process that that gene possibly regulates. So by that, what really do I mean? Let me give you an example. So you've done your analysis and have realized that one of the genes that is most upregulated is NF-kappa B, and you want to test whether inhibiting NF-kappa B could help reverse some of the phenotypes that your cells manifest. Let's see in the literature some of the drugs that have been used to target NF-kappa B. So here in my browser, I will just search NF-kappa B inhibition, and clicking on the first link, I can clearly see that there are drugs that have already been published to inhibit NF-kappa B. Some of these drugs have been listed here, and these have been well researched. So if your cells overexpressed NF-kappa B and you want to see whether inhibiting NF-kappa B will reverse the phenotypes that your cells express, then you will most likely want to pick on these drugs. However, what happens if you have a list of, say, 10 genes or even 100 genes that are overexpressed in your cells? How can you know which are the most possible drugs that you can use to inhibit any of these genes to reverse the phenotypes that your cells express? Now, this is where other biocomputing approaches come in. So in this video, I will show you how to use biocomputation to help you get a list of drugs that you can use to inhibit some of your upregulated genes, or even a list of drugs that you can use to enrich some of your downregulated genes. So here I will basically use R, and I will encourage you to have R installed in your system if you already don't have it, or you can even use cloud computing where they will already have R running in their setup. The R package that I will use here is enrichR. So I will go through its installation and how we can use enrichR to access other external databases and then query our list of upregulated and downregulated genes against those databases to see which drugs can be used to target our upregulated genes or downregulated genes. Installing enrichR is pretty much simple. We simply need to do install packages and then just read enrichR. So I have it installed, and then we just need to activate it by calling library enrichR. As simple as that. So we have enrichR both installed and uploaded in our system. The next thing we want to do is to access enrichR and see which are some of the databases that it hosts, because we will need to query these databases to get the list of drugs that we want. So let's write a simple command to see the list of databases here. We can simply create a variable dps and say list enrichR databases. And basically from our environment, we can click on this to see databases that enrichR hosts for us. So there are a ton of databases that enrichR has, but we won't need all of these databases. We will only deal with databases that deal with drug perturbations, or databases that host data sets where a drug was used to inhibit several pathways or a given pathway. You can see one of the databases here is the drug perturbations from the gene expression omnibus. There are other databases that will be interesting. For example, I like to use the link database, and you can see that we have a lot of link databases here. We have this links L1000 ChemPat app, which helps you to query your list of upregulated genes against the datasets that they have. We also have the ChemPat down, which helps you to query your list of downregulated genes against the links drug perturbation datasets. So there are multiple of these datasets that we will query, but in this video, I will mostly focus on the results that are produced by the links L1000. But at your own free time, you can also check what other databases have to offer. Now, if you carefully see the libraries that are hosted in enrichR, you can see that they follow a given pattern. For example, they have the name part included. And if we go back to the gene ontology library that we saw earlier, we can see that there is drug perturbations included. So basically, we want to filter libraries that contain either the name drug or perturbations with them, because that will be much more specific. Now, we have multiple libraries here, but we only need libraries that are interested in drug perturbations. So to simply filter out these libraries, we can write a simple command using grep to help us scan through all the list of libraries and filter for us only those that deal with drug perturbations. So we can write a simple command as drug dbs and then access the list of libraries within the enrichR using the dbs variable that we already named earlier. So we just type in dbs and then put in the square brackets and then use grep to help filter out the libraries that contain the name drug. I am also interested in whether the library contains the name links, which is one of the websites I am interested in, and whether it also contains the name part that we already saw in the library names earlier. And then I will simply tell grep that whatever I want to scan through is the dbs, but I want to scan through the library names like that. And then tell the grep to ignore any letter case and just return everything that is listed there. So I'll run this. So yeah, I will have the list of libraries that contain the name drug, and then we also have perturbations. Some contain disease, perturbations, some contain links, perturbations. So basically, grep returned for us the list of libraries that contain either the name perturbations or the name drug in them. But we are not also interested in everything that grep returned for us. For example, I am not interested in any aging-related perturbations or even any virus-related perturbations. So you can go through this list one by one and then just pick on the libraries that you are interested in. For this video, I had already created a list of libraries that I am interested in, and I will just copy-paste them in my R environment and then work with them. I will include these documents in my GitHub repo and include the link to my GitHub repository in the video description so that you can go back and also just copy paste this list and work with them. But if you want to have a longer list, you can simply run through this list of libraries and pick on the libraries that you want to work with. For now, I will just work with these ones that I had already selected. So I will just call them selected dlibs. Then copy that and then simply run this and it will give me my list of libraries that I want to work with. Now, the next thing to do is to define your list of upregulated and downregulated genes. So this will vary depending on which systems you're working with. But for the purposes of these demonstrations, I had already curated a list of genes that I routinely work with in our laboratory, and I know that they are upregulated and they are downregulated in our systems. So be sure to work with any list of genes that you feel are important for your laboratory or for the work you are doing currently. So I will just copy paste everything here. I will attach this document in the video description and send it to the GitHub repo. You can access it if you want to use my list of upregulated and downregulated genes. Just paste that there and then simply run both commands. Now, the next thing to do is to query these upregulated and downregulated genes against the libraries that I already have listed here. And I will use enrichR to help me do that. The command is pretty much simple. I will just type upRenrich as a variable and then query enrichR. As simple as that, I will run that. So you can see that enrichR basically queries the list of genes I provided against all these datasets. It's pretty much simple. And then I will also query my downregulated genes. So I will simply copy this and paste it there and just say down enrich. And then here, just change this to down genes and then run that. As simple as that. It just repeats the steps and gives you the results. So up to this point, the entire query has been finished. So what you simply want to do is to access the results that enrichR gave you and see whether there are drugs that you can use to inhibit your most upregulated genes or to enrich your downregulated genes. Now, when accessing the results from the enrichR, you need to be very careful because you need to find drugs that reverse your upregulated genes and drugs that enrich your downregulated genes. So how does this work? If I want to access the list of drugs that reverse or inhibit my upregulated genes, I can simply write head and then tell R that I want upRenrich. You can see upRenrich here that I used to query the drugs that inhibit my upregulated genes. And then I can simply query that using the double square brackets. And here is where you want to be very, very careful because remember, you want drugs that reverse your most upregulated genes. So you want to query the uprich against the links L1000 perturb down. So these are the drugs that repress or reverse your upregulated genes. So I can simply copy this library here and then paste it here and then tell R that I need the top 10 drugs and simply run this. So it gives you a very nice list, but it looks a little bit noisy from my terminal, but you can see that it already gives you a drug called radicizol. I don't know whether that's the correct spelling. And it tells you that this drug inhibits these genes or these proteins, for example. So radicizol can reverse the phenotypes that are induced by these genes such as NF-kappa B1, JUN, and MMP9. We can do this for the genes that were upregulated. I will just need to copy this, paste it over there, and then just change the names here. And then here, I want to be very careful to only want the up library. So you can see how I mix up these two things. So I simply run this, and it gives me another list. And one of the drugs that is listed here is the base 235, which has been shown to inhibit these genes here. So that is pretty much simple and straightforward. You can take this list of genes, look into them in more detail, and then use the drugs actually to investigate the cellular processes that these genes drive in your cells. It's as simple as that. But remember, we also queried the drug perturbations from the gene expression omnibus. So if we want, we can even look at what drugs these two websites give us. So I can simply copy paste these two lines of command and then just change the name from the links L1000 to the gene expression omnibus. Just copy this, paste it there, and confirm that if it is an upRenrich, then I need here to be down. And then simply copy this, paste it here, and then change the name here to down, and then also change the name here to be up. And then run this. So it also gives me the list of drugs that have been shown to target my upregulated genes. And here you can see that it gives me a whole new list. For example, it gives me ubiquinol, and these are the genes that it targets, NF-kappa B1, JUN, FOS, and as well as the HIF1. So it depends with what you want to look at and what you are really interested in. If you want to inhibit MMP9 or even to inhibit NF-kappa B1 or even to inhibit JUN, then you can either go for the ubiquinol or radicizol that was given to us by the links 1000 library. So it's as simple as that. This is a little bit messy for now, but we can make it a little bit clean if it is something that you want to send to your collaborators or even to your supervisors. So you can basically watch our next video to see how we achieve this and how we do other more in-depth analysis of the top hits that we find in our enrichR results. Otherwise, bye for now and see you next time.